So in this section of notes, we're going to be looking at meiosis reduction and variation of the chromosomes. But before we even look at those, I want to really quickly review with you guys the differences in mitosis and meiosis. So simply by looking at this image, you can see a couple of differences right away. At the end of mitosis, you have a goal, and your goal is to produce 2 daughter cells that are both going to be identical to the parent cell. When we talk about identical, they will literally have the same components and same chromosome number. So in mitosis, we will start with diploid cells, and we will end with diploid cells. So they will have the same number of chromosomes. So you'll be producing 2 cells. They're going to be identical to each other and to the parent cell. And then they will also be diploid as well. Whereas in meiosis, you're going to have almost the opposite. Oh, one other thing we should mention about mitosis is that there are only 1 division. You can see you start with 1 cell. This 1 cell will then divide only 1 time to produce the 2 cells. Whereas in meiosis, we have a different goal. In our goals, we are going to produce 4 daughter cells. These 4 daughter cells will be genetically different from each other, or there will be some extreme variations. And these cells will start as diploid, but eventually become haploid, half the chromosome number. And here you can see that our 1 cell has divided once to produce 2 cells. These 2 cells will divide again for a second time to make 4 cells. So we'll have 2 divisions. In mitosis, we will use mitosis to create identical cells, and this is generally for growth, repair, or even reproduction, depending on the organism. And for meiosis, this is specifically going to be to prepare for reproduction. Okay. In mitosis, often we will make somatic cells or body cells. And in meiosis, we often will make our sex cells or gametes. So what we're going to focus on today is, well, how do we get half that chromosome number? How do we get our cell to become haploid? And how are they genetically different from each other? So these are the 2 key points of today's lecture. So the first goal in meiosis is going to be the reduction phase. And in the reduction phase, we're literally going to do that. We're going to reduce the chromosome number. This reduction of the chromosome number is also that main idea of alternation of generations. The fact that we're going to start as a diploid cell, and we're going to produce a haploid cell, and eventually that haploid cell will again become diploid. So this is actually kind of the life cycle or chromosome cycle of the cells. First, we're going to have a diploid whole number cell. This cell will go through meiosis to produce a haploid daughter cell, a gamete. And then that cell will go through fertilization to produce your final diploid cell. Your organism is now complete, or at least its chromosome number is complete. So what are we looking at in this diagram? We should see a couple of things. They do have a couple of phases laid out for you. Interphase, meiosis 1, meiosis 2. We can see that we have 1 cell, and at the end we have 4 cells. So what do we start with? Well, first we're going to start with what's known as a germ germline cell. This germline cell is going to be found in your ovaries or testes, and they can perform meiosis. We mentioned last time that your stem cells do not go through meiosis. The cells that go through meiosis are called your germline cells. These cells will be diploid when they're starting out, just like your skin cells, hair cells, nail cells, brain cells, that sort of thing. So we're seeing a couple things in this diagram. One of the first things you should notice is that we have 4 chromosomes. These 4 chromosomes in the first parent cell, the chromosome number is going to be 4, and they're going to be 4 single cells right now. But what else do we see in this image? A couple of other things that we're noticing here, or should be able to notice. That 2 of these chromosomes are the same size, and the other 2 are also the same size. This first set represents a homologous pair. Okay? In a homologous pair of chromosomes, they will be the same size. They will have the same type of genes, such as the gene for blood type and gene for hitchhiker's thumb. But they may have different versions of the gene. So that's a homologous pair. Same size, same type of information, but there might be some slight differences. And then down here, this is our second pair of homologous chromosomes. Remember that the blue could represent the paternal chromosomes, and the red could represent the maternal chromosomes. So your mom gave you one chromosome, your dad gave you the other chromosome. Together, they form a pair. And in humans, you should remember we have 46 total and 23 pairs. So moving on, what do we see happening next? Well, next we have interphase. And in interphase, you should remember that your DNA is being copied during the S phase of interphase. So in the next image, you can see that our homologous chromosomes are replicated and they have sister chromatids on either side. Another thing you should also notice here is some of the colors have mixed up. So not only has this image gone through S phase, but it has also gone through a process known as crossing over. And we'll talk about crossing over in just a little bit. But crossing over has occurred, and that's why you see the differences in colors there. So next, we're actually going to go through meiosis 1, now that our cell is ready to move on to the next step, and the division of the nucleus can occur. In meiosis 1, this process is also known as reduction division. And basically what happens here is that we're going to divide our pairs. And when we divide our pairs, we're going to reduce the chromosome number. So reduction, divide pairs, reduce chromosome number. So now you can see that in each of these cells, in this second cell we had the number being 4 doubled chromosomes. And then in this next cell, you can see that the number is 2, but they're still doubled. In meiosis 2, sometimes our cell actually doesn't move straight to meiosis 2. Sometimes during meiosis 2, there can be a delay for many different reasons, but the cell can delay this process. But in meiosis 2, the main event that occurs is that we're going to separate our sister chromatids, and that means we're going to separate that centromere region, and that's where the chromatids are connected. One of the reasons why in this phase we don't actually split those doubled chromosomes apart like we saw in mitosis is we actually have different kinetochores that allow this to happen. One kinetochore is just going to move the chromosomes to opposite sides, and we're going to split the number of chromosomes, dividing the pairs. And then in the next set, the kinetochores will actually split those chromatids apart, and so you have your separate sister chromatids. So how does the stages of meiosis allow for the reduction of the chromosome number? Just as a bit of a summary here, we have meiosis 1. This is where we separate our homologous pairs. In meiosis 2, we're going to separate our sister chromatids, so the replicated chromatids. When you're looking at these two images, we have an image of meiosis and mitosis. What you want to be able to do is think about what errors can occur during this separation. So what kind of errors can occur, and what would be the results of those errors? And hopefully you can think back to Bio 1, or even look up for a minute what are some results of the chromosomes not separating properly, and think about where and in what phase would this occur. So just to talk a little bit about these images, what you're seeing at the top here in this first cell has actually, or is going to undergo, prophase and pro-metaphase already. Notice that the chromosomes align at the metaphase plate. When chromosomes align at the metaphase plate, we'll also need the metaphase checkpoint to occur here. Your daughter chromosomes will separate. When your daughter chromosomes separate, that's going to be anaphase. And then this section is actually showing you telekinesis and cytokinesis all at once. However, in this picture, what we're seeing here is that we see metaphase 1. And the reason why you know that this is metaphase 1 is because our chromosomes are actually going to be lining up as a tetrad. Tetra usually implies 4, which means our homologous pairs will line up.
Sierra Bodrick, Placenus Ellis in Student Services, Eric Robles, DeKayla Shukershad, Stephen Sings, Maria Smith, Tyvon Smith. Please see Ms.